Assalamu alaikum. In this lecture, we will cover the remaining MCQs from the paper, question paper number 12, March 2022. So let's start. The statement describes the structure of a polysaccharide found in the cell wall of the certain plants. The polysaccharide is composed of two different monosaccharides. No, it can be made up of the more than two different monosaccharides. The monosaccharides are joined by the alpha 1,4 glycosidic bonds. Yes, it is always joined by this bond. The polysaccharide contains pentose sugar. It is not necessary. It can be hexose, triose, and some other as well. So option D here is the right one. The general formula for the saturated fatty acid is this. What of these fatty acids are unsaturated? We have to find out what are unsaturated. What does not fit in this formula will be unsaturated. So if we will find out, then we can say that 1, 3 and 4 do not fit in this formula, so they will be unsaturated. So option C here is the right one. What enables the triglycerides to perform their function in the living organisms? Triglycerides have hydrophobic and hydrophilic regions. Yes, but they are mostly required in the cell membranes, not in all spaces. So triglycerides have a high ratio of carbon to hydrogen bonds than the carbon atoms. Yes, there the carbon and hydrogen bonding is very higher than that of the presence of the carbon atoms. So carbon hydrogen bonds can play a very important role in the different functions. So for example, in cell signaling, in maintaining the fluiding of the cell membrane, and many others. Hydrolysis of triglyceride releases metabolic water. It does not release water. So we can say that option D here is the right one. The diagrams show the three examples of the different bonds. Here it is the hydrogen bond. It is the cyanide bond. Or it can be said to be the amino bond. Amide bond. This is the sulfide bond. So, which bond can hold together the quaternary structure of the protein? Quaternary structure of the protein requires hydrogen bonding and sulfide bonds. It do not require any kind of the amide bond. So option C here is the right one. Which row correctly describe the primary structure, secondary structure, tertiary structure, and the quaternary structure of the some proteins? So we have to read all the options and find out the right answer here. Primary structure determines the folding of the polypeptide. No, it does not determine. So there is no need to read the other options. So because one option is wrong, so it will outrule the whole option number A. So let's move towards the other. Primary structure define the order of the amino acids in the polypeptide. Yes. Usually form, secondary structure usually form immediately after the polypeptide synthesis. Yes, it can be. It is held together by all type of the bonding that occurs in the proteins. Yes. By all type of bonding, only a few bondings can be present here. Quaternary structure found in the globular proteins such as the hemoglobin and never but in the, yes, it can be. So it also outrules here. It involves a covalent bond only. Yes, it involves. Secondary structure involves the interactions between the hydrogen and oxygen because there is a hydrogen bonding involved. Yes. Tertiary structure is essential for the function of the enzymes and the receptors. Yes. Mostly enzymes and the receptors they are the proteins and they have the tertiary structure. Quaternary structure is formed when the two or the more polypeptides join together. So option C here is the right one. What relationship could be investigated using a calorimeter? The calorimeter uses the light intensity and the temperature, so they will be the mostly discussed here. The effect of the light intensity on the rate and with the solution of the light sensitive dye changes from the green to colorless. A dye which is used in this is green in the color, and the light intensity, the intensity of the light will have a significant effect on the color changes. So it can be investigated using a calorimeter, and yes, it can be. The effect of the temperature on the rate of the breakdown of the cell membranes in the tissues with the pigmented cells, such as the beetroot. Yes, effect of the temperature can also be investigated using the calorimeter. The effect of the pH. No, we cannot determine a pH because for this purpose we have required a pH buffer, but we do not use a pH buffer in the calorimeter, so we cannot find out any effect of pH here. The effect of the light intensity on the rate of the change of the skin color of the lizard, but we cannot study the lizard with the calorimeter. So we can say point 1 and 2 are the correct. So option number B here is the right one. What transport mechanism does not require a concentration gradient to be present in order to take place? Exocytosis. Exocytosis do not require any concentration gradient because it is just the movement of the water or some other molecule out of the cell. Facilitated diffusion and osmosis and transpiration, they all require the concentration gradient to take place. So option B A here is the right answer. High concentration of the ethanol disrupts the cell membrane structure by the denaturing the proteins and increasing the separation of the adjacent phospholipid molecules. As a result, the cell membranes can decrease in thickness and become more permeable. Because the thickness and the more porous the membrane will be, the more will be its semi-permeable. But when its thickness will decrease, its pores will become more open and it will become more permeable. So more molecules can pass through it. Ethanol can also pass through it. In the case of the yeast produce ethanol in the many metabolic processes. So due to the repeated production of the ethanol, the yeast cells become tolerant of ethanol. So their cell membranes are not degraded by this. Which statement correctly explains the response to the ethanol that could account for the increase in the tolerance of the yeast cells to membrane? We have to find out the particular reason why the yeast cell membranes are tolerant to the ethanol. Decreasing the ratio of the saturated fatty acids to unsaturated fatty acid within the cell membranes. Helps to prevent the tails of the phospholipids on the one side of the bilayer from sliding past the tails of the phospholipids on the other side of the bilayer. Yes, it will have to prevent it so that the structure of the cell membrane can be maintained. Increasing the proportion of the palmitoic acid and to the oleic acid to the phospholipid bilayer increase the fluidity of the cell membrane. The fluidity of membrane is not related to the proportion of particular fatty acids. It is related to the structure of the cell membrane. Activating a cell signaling pathway triggers the unfolded membrane protein response in the endoplasmic reticulum. The unfolded protein responses poses the protein synthesis and initiates a cell death in the yeast cells with a high proportion of the misfolded proteins. But cell signaling is not related to the cell death and the protein synthesis. The many other processes are involved in this. Increasing in the proportion of the ergosterol in the cell prevents the accumulation of the polar molecules. But ergosterol is not involved in the accumulation of the polar molecules like ethanol in the cell. It is related to release them. So option A is the right answer. In an investigation, a plant cell was placed in the pure water. The initial rate at which the water molecules entered into the cell R was greater than the initial rate at which the water molecules left the cell. In the second investigation, a plant cell of the same type was placed in a solution with a water potential equal to that of the cell contents. Water potential is same in the solution as that of the cell, which is placed in that particular solution. So, what we investigated after a period of 5 minutes, what will happen? The water molecules will enter in the cell or will leave the cell in the same ratio because the water potential in the both solution and that of the cell contents is same. So we can say option B here is the right answer. The protein P53 is produced in a cell in response to the DNA damage. When the DNA is damaged, then the P53 protein is produced. This protein stops the cell cycle for a short time just before the DNA is replicated, so that the DNA can be repaired. So, at which phase the cell cycle will this stop occurs? Because DNA replication appears in the S phase, so before the start of the S phase, the DNA has to be repaired. So we can say that that the P53 stops the cell cycle at the G1 phase. So option B here is the right answer. I will finish my lecture here. Till my next lecture, stay tuned and Allah Hafiz.